So yeah, if you go to the Orchestra app, the link that I put in the chat, you will be prompted to log in if it's the first time.
Yeah, to register, I should have to register. So I don't know if it's time for the registration right now.
Well,
by the way,
you can register. You can log in with Google.
Okay.
And then you will see all the workshops that are in the system, and you can search in the top right corner for Spatial Experiment. And you will see that, and you launch. No, you launch.
Okay.
No, go back. You launch, and it will be an RStudio instance will be created, and you will be prompted to log into RStudio, and the username and the password are RStudio.
Yeah.
So if everybody wants to do that now, so that then we can start, and everybody will be ready.
Yeah, I can. My part is an introduction, so.
Right. So while RStudio loads,
yeah, exactly.
Dario, you can go on. Yeah.
Welcome everyone, and so today we are going to talk about this spatial workshop. It's divided in three main parts. So the first one is an overview of the technologies for spatially resolved transcriptomics. The second part will be hosted by Helena, showing the new SpatialExperiment class. It's more an hands-on section. And the third part is took by Lucas, showing the ST data package and the GG Spavis package. The first one is a package for it's an experiment hub package for data sets, spatial transcriptomics, and GG Spavis is for the visualization of these spatial data. So here you have some additional resources, but and here in the top upper this corner you have the three parts. My part is the introduction. So what do we intend for spatially resolved technologies? We refer to all those technologies which provides one or more omics expression data and also their spatial information. We are going to focus on spatial transcriptomics. So the main omics is the transcript. Are the transcripts, and we are able to detect transcript quantification and their spatial coordinates into an under-study tissue. We distinguish two main families, so the molecular-based and the spot-based until now. And for the molecular-based family, the main idea is to use multiple temporal snapshots to detect the transcript locations, and they can work at cellular and subcellular level. We have SecFish, SecFish Plus, MERFish, and other technologies. The main differences within this class are due to the way to detect the transcript coordinates and the number of the detected transcripts. So for the SecFish and the SecFish Plus, the main difference is that the SecFish Plus is an update of the SecFish technology, but the idea underlying this technology is the same. So they use temporal barcodes to detect in multiple hybridization rounds in combination with image microscopy and the FISH approach. So fixed one temporal barcode. So the temporal barcode will be our cell identifier, we can say, the sample identifier. And inside this temporal barcode, we have multiple hybridization rounds where the transcripts are detected, are localized inside our barcode, our temporal barcode. And the end of the hybridization rounds, let's say that here we have n equal to 6. We have a barcode key for each of the transcript. So for this particular transcript, we will have these legend of colors to identify this specific transcript. More in detail, if we have, for example, here, these 3 hybridization rounds, in the end, our barcode scheme will be for the mRNA1, yellow, blue, yellow. And as you can see, you can identify the mRNA1 3 times at the end of the process. So in the end, you will say that mRNA1 will have 3 counts for this specific barcode, for example. You can apply this to a tissue slice, for example, and you can zoom with a microscopy, of course, into an interesting area until to reach the desired resolution to do your hybridization rounds. What I was gonna say is that for the SecFish plots, we are able to detect thousands of transcripts. So this technology is comparable with a whole transcriptome technology. While the spot-based technology, with this other family, we identifies all those technologies that are, as a reference, the spots that are uniquely identified with barcodes, and the coordinates are associated to these barcodes. And for each of the spot, we have the transcript abundance. Main differences within this family are due to the spot size and the spots reference protocol used. For this workshop, we are going to focus on the 10x Visium technology. They use a chip, a spot barcoded chip, so you already know how many spots you have on the rows and on the columns, so you know the coordinates of the spots. You apply a tissue slice on the chip, and while we take the image of the tissue on the chip with a microscopy, at the same time the chip starts the cDNA amplification inside each of the spots. So at the end, we will have the spatial information associated to the spot and also the whole transcriptome amplification inside of each spot. This is a big difference between these kind of technologies, so the spot-based and the molecular-based, because in this case, we have the cDNA amplification, while in the other case, we don't have it. Okay. To analyze this data, we came up with this SpatialExperiment class, which inherits from...
1 second, there's a question.
For sure. How cells boundaries determined? I'm not sure about how to answer this question, but I think that it's up to the zoom resolution that you apply with your microscopy. But I'm not so deep inside this technology, so from the bench side. But I suppose that it's
and I think I'm not sure, but typically with microscopy based, they would do a DAPI staining of the nuclei, maybe, or there would be some kind of membrane staining and then segmentation. So like there's a separate staining for that, maybe.
I think also is also providing the answer to that using DAPI and mRNA composition. So this was another question in the Q&A. So it was an A to the Q.
Okay, that's good to know, by the way.
I think the answered ones for you.
For the commercial company, I don't think so. So at the moment, I didn't saw any company performing this. The commercial company performing this technology, at my knowledge, but I don't know. So in the end, we came up with this SpatialExperiment class. which you are going to see more in detail during the next session. So we stored the spatial coordinates. So because it inherits from the single cell experiment, it has the same structure. And we are still under the development of this class because we are facing several issues, in particular because of the two kind of technologies families. And so for the spatial coordinates, we decided to store them inside the cold data structure, and Helena came up with this other data structure, the image data structure, useful to take care of the samples images. So in the end, this new class is able to handle multiple samples and multiple images for each sample. And it also stores the image itself if you need it. And for the Visium, you will need it. We decided in this way in the end. And so we have multiple attributes and accessors for these structures. I'm not wasting time about describing them because we are going to look into them during the hands-on. Just an extra resource that I want to show you is if you are interested in a SecFish dataset, you can download a dataset of SecFish from the single cell multimodal bioconductor. This dataset is composed of two assays, a SecFish data assay and a single cell RNA-seq assay. And all the information about this dataset are collected inside this repository at this link, and you can download and install the package with this code. So if you have any other questions, otherwise we can go ahead with the hands-on.
So I hope those who are interested in typing along have their session open. Can you see my RStudio? Okay, good. I'm keeping also this open for my own reference. So we want to have a look at some Visium data and especially the the spatial experiment class. And in the interest of time, we could read in this data manually. There's quite a bunch of data that we will see that goes into this object, but instead we'll be using a function to do that is kind of similar to read 10x counts, for example, to read in droplet-based 10x data. So we have a similar function to read in the Visium data. And the reason for that is because it comes in a very fixed format. So that's like outlined here for every sample. Running the Visium pipeline gives us this HDF5 raw and filtered count data. Then also a directory with the same data in like sparse matrix format. Then there's the spatial subdirectory that has a couple images. These are described also on the 10x website what these different images are. There is the tissue positions list CSV, that is our spatial coordinates and some other information. And this JSON file that are scale factors to adjust our spatial coordinates to the image resolution that we're interested in plotting, because otherwise there might be a mismapping between these. So in our example, I'll try to live code this as far. Oh man, this is terrible. We have provided some example data in the Xdata 10x Visium inside the package. So I'll just specify that here. And there's two subdirectories in there for two samples, and these are from the 10x website. So these are just two consecutive mouse brain something sections. So I'll just save these here as sample IDs. So these are our two samples. And then the complete paths to the different files, all of this code is of course here, are are here. So these are our two complete paths to the data. And then we can look at what's in these. Let's do that. So we only provide a subset of all this data. So typically you would have a bunch more. We just have the HDF5 raw counts and this spatial subdirectory, which contains our scale factors, a low resolution image just because it's small. The full resolution, high resolution images can be up to like 0.5 gigabyte, and our tissue positions. And as I said, we'll be using a function to read these in, and that's the 10x read 10x Visium. And what that wants is these file paths, the names we want to give on our samples. I stored these here as sample IDs. We want to say which images to load because there can be a bunch of images. So here I only want to load the low resolution image. I'll also say load equals true to say that we actually want to read the images into memory. If this were false, the images would be stored but not read into memory. And I believe
you've got a typo in sample IDs.
I actually didn't load the package, so I need to also load the package. Yeah, so sorry. Of course, we need to load the package, and then this should work. So I'm calling this VE for Visium experiment, and this looks a lot like a normal single cell experiment. We have about 30,000 features, about 10,000 observations. These are spots, not cells. We can look at the spot metadata we have. So we have our barcodes, sample IDs, the X and Y coordinates of the spots. And these are pixels, so they're integer values. We have an indicator if a spot is in the tissue or not. This is output by 10x. So they have like a method, I guess, with a staining where they see if a spot was mapped to tissue or not. So we don't have to do this. And then there's also this array row and array call that tells us the position of the spot on the chip, because we have this grid with a bunch of spots. Of course, as Dario pointed out, we have a couple of accessors like the spatial chords accessor to really retrieve the spatial information only. And this comes from this tissue position list. And just, this is not in the demo, but just to see what that looks like, I am going to subset one sample. So this is just the section one sample. And we can then plot, for example, the X and Y coordinates. Oh. Go. And it's just a grid. So that's not very useful. Let's color that by tissue. And then this is what our spots we can see. And then the spots that are.
Did you lose Helena?
I think we lost Helena. In the meanwhile, probably you can take a chance to pick up on some of the questions. I think there was one from Friederike Dunda asking whether Elena could put the read command in a script or paste it in the chat. I think it's all in the vignette of the rendered package down package of the website, so you might be able to catch up with that.
Yeah, so all the code is on that compiled website of the workshop. There's at the top there are 3 tabs for the 3 sections, and the one that Helena's working on right now is the spatial experiment one. So all of the code that she showed is in there.
Maybe Lucas, can you put in the chat feed of that instead of the Q&A so that it persists throughout the workshop, and then you can just for you, Friederike, you can just click on that. And it looks like we still have...
I'm doing it now.
Okay, perfect. Thank you. And then
it, right?
just checking in what is still unanswered. I think it's regarding a 4-cell segmentation from also the command. I think we can mark that as answer, or can you see that in the Q&A, guys?
Oh yeah, the load true false or the other one.
It depends whether you want to pick up on the most recent ones or the
latest,
or on the.
Yeah.
I think the segmentation we talked about, so we move to the next one.
Okay.
Do you want me to relay the questions to you?
Yeah, thank you.
All right, so starting from the most upvoted. Well, this is not really a question, but there's a bioarchive link for cell segmentation.
Yeah, thank you.
Yeah. So, yeah, there is also a paper from the Karchenko lab to refine segmentation post-hoc. I guess this is really useful. Then in terms of questions, what does the load true false in read fun do? Is it like on disk or memory?
Yeah, it's if you put a load equal true, it loads the image. So it loads the image into the memory. So you can otherwise you can leave it to false and it stores just the path or the reference, because we can load also URLs with this function. So we leave the decision to the user if they want to load or not the image, which is stored inside a grob object.
Mm-hmm. And so there's a question by Michael Stadler. Samples are row bound in the assay. What was the reason they are not represented as different assays? I think we talked a lot about this, right?
This was something that we had quite some discussions about. And the reason we decided to put it all in one big assay in the end is that this lets us reuse all of the packages like SCADA and SCRAN from single cell experiment work. So we can do normalization and so on using those existing methods. If each sample was in a separate assay, then we would have to rebuild all of that infrastructure to look more like the FS supply in flow cytometry. I think it's better for us to keep it as one big assay.
Yeah.
Then there's a question by Alsu Misarova, and the question is: do the spots in Visium overlap physically?
Yeah.
No, so they're 100 micrometers apart and 55 micrometers.
Yeah, there are some of the spots that overlap physically and the other ones not, right?
No, it's just a big grid.
Okay, but you can define the spots that are on the tissue and the other one that are not.
Yeah.
Yeah, no, I think the question was more they overlap with each other, but they're not.
Oh,
they're on a regular grid.
It's a hexagonal grid with a very regular spacing.
Okay, sure. No, of course not.
Okay, so I think in the interest of time, since it seems that we lost Elena for good.
Yeah, Lucas, maybe.
Lucas, maybe you can share your screen and go to the spatial viz.
Yeah, that sounds good. So and the rest of Helena's example there is in that HTML there. There's quite a bit more where she steps through the object structure, so you can go through that.
Wait, before you do that, there's a comment by Mark Robinson. They do not overlap physically, but can RNAs disperse down into neighboring spots?
Yeah, maybe a little bit. This is something that I think is not really fully known yet to what extent that happens. I know people are looking into this, but yeah, I think there's no definite answer to that. I think it's minimal, but maybe turns out it is a bigger problem than we expected. I'm not sure. Yeah, we're going from the assumption that it's minimal.
Great. Okay.
Sweden is still trying, but it's. I don't think it's really.
Yeah, I think we, Lucas. I think we have 15 minutes left or so.
Yeah,
maybe
that's
20.
fine.
But
my examples are not too long. All right. So here I've just gone back to the website, and I'll just show you again that. So at the top here, so you can see my screen. This is
yes.
Chrome. I've got the BIOC Spatial Experiment Workshop website up. There's some information here, how to install it. Then in this articles tab up the top, they're actually out of order. So the first one is the one that Dario started with, the Introduction to Spatial Transcriptomics. Then number 2, the Spatial Experiment class. This is what Helena was going through. So this read 10x Visium function to construct the objects. And then the rest was going through more examples of what the object structure looks like. So then in my one, that's the ST-data package. So here, what we've got, so we've, alongside the spatial experiment class work, we're also building up a small package which we'll submit to Experiment Hub shortly, which has some examples of nice small data sets in the spatial experiment class that we can use in our examples. And that's currently on my GitHub. There's a link here. Yeah, and that's installable with the current version of Spatial Experiment. Now, currently, we've got 2 data sets in here, both from the Visium platform. The first one is this mouse coronal one, which is quite similar to one that Helena was using in the package as well. This one is a coronal section through the mouse brain, so a different axis from the other one. And then the second data set is human dorsolateral prefrontal cortex. That's one small slice out of a human brain sample, showing the 6 cortical layers plus white matter. That's from our paper that's on bioRxiv. And also that whole data, so that's one of 12 samples in that paper. The whole data set is available through the spatial LIBD bioconductor package that Leocadio Torres has put together previously for the paper, and which Brenda Pardo talked about the other day as well on updating it to the spatial experiment. Now in the ST data package, so if you're working locally, you can install it from GitHub with install GitHub. Remotes package, if you're working with the Docker container, it should be there. Then I'll step through a small example now showing how to load the data. And again, all the code is here in this HTML if you want to refer to it again later. Now I'll need to, I think I need to share the different screen. Okay, can you see my RStudio? So this is the same page that I was showing before. Now it's the RMD version. So first we load the package, both spatial experiment and ST data. And then for now, we've just got this simple function that load data with the name of the data set, which while it's on GitHub, this gets it from a Dropbox link, but that will be updated to use the standard Experiment Hub code, which then you've got sort of 2 or 3 lines to load it from Experiment Hub. We'll update that example to show how that's done. Then when it's loaded, then yeah, we can look up the object similar to what Helena was doing before. So it's a spatial experiment class. This one has 32,000 features, 2,700 spots. Got a counts assay in there. And if you have worked with the single cell experiment class or the summarized experiment class, you'll see that the difference is these things that Dario was talking about earlier on the spatial coords and Helena, sorry, on spatial coords and image data. So these are the new pieces for the spatial experiment. And with these lines of code here, we can have a closer look at some things like the size of the object. Assay names tells us what we've stored in here, which is just the raw counts. Row data. Row data is the information on the features, the gene IDs and gene names in this case. Col data is the entire everything that's in col data, which includes both spatial and other information about the barcodes, which again, if you're comparing this to a single cell experiment, this would be the cells, but now it's the spots in the Visium context. So we have the barcode IDs for each spot, then array row, array col, which Helena showed on the location within the Visium slide. Pixel col, pixel row in full res is the same thing, but scaled to the image size. We've got a sample ID, and then the X coord and Y coord, and in tissue, that's the part that's also available through spatial coords, which is used by plotting functions. And the image data, this is the actual image information, GROB and so on. So now we'll make a simple plot to show what this looks like. We've got this other package called ggSparvis, where we're planning to collect a series of visualization functions. That's also on GitHub for now. right now we've just got a couple of simple visualization functions in there. Again, if you're working local, you can install it from my GitHub with that line of code, then load it. And this function, plot spots, should make a plot of just the spots over tissue. So that's using, in the spatial coords accessor, there's the X coord and Y coord and the in tissue column. The in tissue column tells of which of these spots have been identified in the space ranger output as being over tissue. And so here we're just showing the spots over tissue and not all the background ones. And this is the mouse coronal section that I mentioned. So that's a hemisphere of a mouse brain if you've sliced it vertically on the coronal axis. Now, as you can see, that's not a hugely interesting plot. That's good for quality control. Just check that your data is actually loaded correctly. It's not completely messed up in some way. Now, to do a slightly more interesting plot, we'll load the other data set, the human DLPFC. I'm calling that SPE again, Spatial Experiment. Let's got it from my GitHub. Then make the same plot spots SPE. That shows the orientation of this piece of tissue. And of course, the big difference here biologically is that the human brain is so much larger than the mouse brain. So this is, I don't think we actually mentioned, so the size of the VCM slide is, I think it's 6.5 millimeters by 6.5 millimeters. So that's the dimensions of these tissue images. So in the human brain, that's one small piece, whereas in the mouse, it's half of the brain. Okay, and now in that object for the human DLPFC, from our previous work in that paper, we've also got ground truth cluster labels for the layers, the cortical layers, which we've stored in that object as well. If we run that code, it colors them nicely by those layers. So the cortex biologically is divided into 6 layers, and below that is white matter. And in this plot, we're showing the white matter with black, and the other multiple colors are the different layers, which in these ground truth labels, these were manually annotated by our collaborators. And now, in the last few minutes, we've got an exercise that people can try, and if we don't get through it all, also continue later if you want to from the code on the website. So what we wanted to do in the exercise is try to modify this plot to show something a bit different, to show a continuous color gradient instead of cluster labels of total UMI counts per spot instead of using those ground truth cluster labels. And I've included some hints for how to do this. So using that same object, SPE, have a look at it, inspect it, look at the different pieces, row data, col data, spatial coords, and so on, and see where that information might be that you need. And then have a look at that plot spots function, what its inputs are,
so
where you
may
could
I
provide that information.
That
Yep.
people can try the exercise on their own, maybe after the workshop, and we use these last 10 minutes to go back to Elena's part, so that we don't miss that part.
That's a good idea. That's a great idea. So this is actually the end of my materials. So so there's this exercise here. You can have a look at that on the HTML on the website. If you are looking for the solution, you can dig through the GitHub. It's in the RMD, the solution. I'll just tell you that in advance. But of course, it's better to try the exercise first by yourself. So just look at the HTML where the instructions are. But then if you're looking for the solution, go and find the RMD version on the GitHub, and you'll find it there at the end of this script if I scroll further down. I'm just looking at the top of the list now. So Mark has asked, the layers are thick in that each spot is multiple cells. No, the layers are really thin. So they're approximately 1 cell thin. But yes, it is. Yeah, it's possible that you might have some overlapping behind them. In our data set, I don't think we had that problem. They were, so in the human cells are pretty large. So there wasn't that sort of, cells behind each other. But in mouse data, that certainly might happen, yeah, because they're smaller. Kelly said I cut out after coloring cells. Okay, sorry, yeah, so the rest of the code is there in the HTML if you want to continue with that exercise.
I don't know if that was me or if that was for you. I mean, I was just going to continue. Now, Lucas already explained a bunch of stuff, but so far we've looked at the spatial data. And the second thing is this image data that is new. And here, essentially, each row is an image entry, which we've also seen in Lucas's example. He had 2 images for 1 sample. Here we have 1 image for 2 samples. And then the scale factor would tell us how to adjust the X and Y coordinates to the image resolution. So if we had a full image, full resolution image, this would be 1. But the full resolution image can be 500 gigabytes big. So that's why these images here are much smaller, and we have to adjust the things. The images themselves are stored in this data column as spatial images. So this is a class we implemented that can store a URL to source the image from, a path if the image is sitting on our computer, and the grop. And this is actually allowing us to plot the image using the grid package or ggplot. And in this case, both our images are loaded, so they're physically like in memory and in the object. Just to maybe demonstrate, we could unload these images. I'll set this image ID and sample ID true, just says all samples, all images. So if we do that, and again look at our images, these now say not loaded, grop is NA. So the grop has been removed, and if we store this object, it's very lightweight because we're just storing paths. We can revert that with load image. So this loads back our images into memory. And then actually my favorite part that I wanted to show was we can also add new images. So in the workflow here, there's a URL from an image from Google. So I'm copying that here. So this is some image. We can say add image from this URL. We need a scale factor. I'll just do 1. And we want to add this to the section 1 sample, and we give the image an ID. So this is kind of, you can look at the details of the function, but essentially we want to add this new image entry. So now we see here there's this 3rd image now, and that's the image, and we can plot that. So we can say grid draw image grop, or actually let me save that image. So it's the third spatial image, and we can draw this image, and then we have a picture of a cute dog. Yay! So no, sorry, this was more of a fun fact, but the point is you can store images in some kind of external resource like a server. You can add them to the object from a URL or from a path. You can visualize them, and in the end you can remove them, load them, unload them, and all these kind of things. So that's kind of the idea of this image data frame. And I think we're done. Maybe just a last word, or is there
There's a couple more questions as well. Maybe
okay.
There's one I wanted to answer there from Aiden on protein and VCM. So yes, VCM is working on a new platform for proteins as well. I think, yeah, that's coming. We don't have that yet. Yeah.
No, but I saw that there is 10x spatial proteomics, which gives you also the transcriptomics.
It'd be antibody-based protein plus the transcriptomics.
Yeah.
They've either just released that or they're working on it, similar to CiteSeq, but on spatial, I guess.
There were some comments from Mark about the layers being thick, in that each spot is multiple cells and the cells are multiple spots thick.
Yeah,
but probably
we answered that. Yeah.
So okay, and then David's comment about how do you imagine sharing these objects in example Experiment Hub, will the URL point to the AWS bucket?
In Experiment Hub, you put the actual formatted spatial experiment object, and then there's this make data script that shows how you create it from scratch. So there, in that make data script, we would have the link to the raw data where it came from, whether that's AWS or somewhere else, depending on the data set. Yeah.
Okay, so is the idea that you never download the image unless you want to, or would it download it from Experiment Hub automatically and then point to the file on your disk?
The small one it'll include, but not the 0.5 gigabyte one.
Yeah, like I think in the data that I'm trying to make right now, I load the low resolution and I save all the others as paths or URLs.
Yeah, same.
So they're like in the object and accessible, but they're not loaded.
Because the low resolution one is like a couple of megabytes or something, whereas the full resolution can be up to literally gigabytes. So you don't want that in there by default.
Cool.
Yeah.
There was another question from also Ms. Roba. Not familiar with Vision much, but I guess the difference with single cell would still be in the fact that one spot combination of overlapping cells, as in the spot could lie on the border between cells, might be relevant for cell-cell interactions.
Yes. So there's a lot of work going on with from other groups on how to deconvolve cell types within spots. If you have a tissue type where there's lots of cells per spot. Following on from the bit that Mark asked earlier, so in our human one, we had on average, I think it was 3 cells per spot, but there was a range from 0 to about 10, depending on where in the tissue it was. So ours was close to single cell, but in the mouse data, I think that's around 50 cells per spot. So then things like cell type deconvolution are important. There was a nice paper from Irizarry group, Cable et al.
Okay.
And also the base space method was doing this from Gotardo's group.
Okay, sorry, no problem. Also, it's not about. So I mean, the conversation is not so. It can't be so really easy with the Q&A tab. There's another one from Mark asking whether there are any Experiment Hub datasets with both spatial and dissociated single cell RNA sequencing on the same tissue, maybe from adjacent slides.
Not that I'm aware of on Experiment Hub.
I don't think so.
Yeah, not yet.
Okay. In the single cell multimodal, says Aidin.
Yes, thanks, Aidin. Yeah.
Okay, maybe Aidin, if you want to post the link to that in the feed.
Well, I mean, so it's Dario's package, so Dario should know.
Yeah, that's the one that Dario mentioned that right at the beginning. That
Yeah, by the way, it's in the last introduction. In the last part of the introduction section.
Yeah, so I think it's slightly different from what Mark was asking, because in that case it's the same. I mean, it's it's independent data sets, right? It's seqfish and single cell RNA-seq on the same kind of region of the mouse brain, but done in different labs, right? Do I remember correctly, Dario?
Yeah.
So it's not adjacent slices or the same mouse.
And there are no adjacent slices there, I think. So there are different experiments, and the group of the Beers workshop made all the preprocessing of the data to be matching the two data sets. And then we stored the already processed data sets inside the single cell multimodal package, the experimental package. And you have seqfish and single cell RNA-seq, not Visium.
There was just a general comment from Mark for the excellent, compact, and efficient workshop, which I can just subscribe to as a comment. And say, despite connections issues, we saw the Pomeranian dog, so the universe is back in equilibrium, I would say. Great talks and great discussions.